Hello everybody, my name is Brendan Genaw. Today we're going to be talking a little bit about nature and nurture. So a little bit of background information. I am a biology major here at the University of Michigan, and my honors thesis is with the Deuce Lab, and we study how sugar consumption affects our DNA and our genome. So a little background information. This is me in 9th grade. I used to run cross country. I still love to run. And this is the first time I felt the power of sugar on my body because we decided to drink Mountain Dew right before a run. It was not the best run, and I blame that beverage for it. But one interesting thing that I noticed was one of the guys on our team always drank Mountain Dew before he ran. It didn't seem to affect him at all. And it really started to wonder, like, what makes someone a fast runner? Is it something you're born with, or is it something that, like, getting good sleep, eating a good diet, that really helps? And that's where we get into this debate, which is the debate between nature and nurture. So on one side, we have nature over here, which really looks at your DNA, which looks at what you're inherited, which stuff that you're just given, you don't really control. Then we have nurture, which is over here, your environmental influences, your experience, your childhood, and your development. We see this a lot in the media on topics such as intelligence. Are people born geniuses? Is this something that through your experiences you develop and you grow your mind? Stuff like personality. Sometimes people say you get your temper from your mom or your dad. Is that something that you can actually inherit, or is that something that maybe parenting really can control whether or not kids act that way? We also see this with sexuality and health. So with health, for example, some people never went to the gym in their life and have great bodies. Other people go to the gym every day and still struggle with their weight and what they want to look like. So this debate's been going on for a while, and one big thing we see is it's normally two different sides. So you have a lot of people say this outcome is from nature, this outcome is from nurture, and they're battling each other. However, that's not necessarily the case. So recently this new field in biology, it's kind of a hot topic, is epigenetics. And this actually looks at how our environment can influence our heritable traits. So how our environment is actually working with our DNA. It's not one over the other to create an outcome, it's one with the other and an outcome is formed. So a little biology lesson. With inheritance, you are made up, your DNA, half of it comes from your mom, half of it comes from your dad. Your DNA, that's what it kind of looks like. We call this a double helix structure. The blue and the red are backbones. So this is your DNA structure. What's really important is this yellow and this green right here, and that's the code for your DNA. So your body, your DNA acts as your blueprints for everything. Whenever your body needs to do functions, it consults your DNA and it reads that code. And that code is something given to you by your parents. The code is static, so once again, these code, you're born with it, it stays relatively the same. However, the DNA structure is not the same. Your cells can actually change how your DNA is within the cell to make things easier to access. So what do I mean by this? We're going to look at this kind of as a metaphor. So if you think about your body as a business, as an office space, like every office space has a bunch of different cubicles where work gets done. Your body is comprised of cells where a lot of work gets done. We can kind of think every cell has DNA in it. And once again, half from mom, half from dad. This DNA is in every single cell. They all have the same amount of DNA and the same information in it. So for example, in your stomach cells will read the DNA to make stomach acid for digestion. Your eye cells, they don't make stomach acid, but they still have that DNA, that information in them to do that if they wanted to. So if we look at our office analogy, that'd be kind of like having every cubicle has this huge protocol about what you need to do in the office. So if you work in the financial department, even though you might never work with the environmental lawyers, you'll still have a section on there about environmental law and contacting those lawyers. So you might never access it, but it's on your desk. And this causes a problem, because if you have all this information, what do you do with it? So in an office, you will probably have file cabinets, and you can file it away and organize it. In our cells, we have these things called histone proteins, so these blue little circles, and your DNA is coiled around them. So just like in an office or on a desk, if this is going to be our papers and our DNA, what we're going to access, things that we put on top of the desk, stuff we probably access very frequently, and things we put in the very back is stuff that we really don't need to access. Our DNA does the same thing. Certain cells will decide what it needs to access and might coil it tightly around it and be like, I don't need to access this, we're going to store it in the back. It might leave it loosely around these histone proteins so it's easy for the cell to get to. Now, this is kind of what's cool. This is where new information comes in. There's this thing called epigenetic modification. And what this is our body's way of marking certain pieces of our DNA to make it much more accessible. And this is how our DNA interacts with the environment. So for example, if I have this stack of papers and I want to really get to this green paper very frequently, I can put a sticky note on it and you can access it pretty easily. Your body can also do this. It can also make things more difficult to access. So if you have a red sheet of paper in this stack and you want to make it a little more difficult to access, you don't use it very frequently, you might put a clip on it. So that way it's a little easier, but it's harder to get to. So these are known as epigenetic modifications. And this is what our body does in response to our environment. So once again, the actual material is not changing, nothing is being rewritten, but the way that we access it, the way that we get to it is different. So to summarize, our environment uses these epigenetic modifications to alter our DNA. And this can actually be seen in offspring. So this is heritable. So what? Why does this matter? Okay, well, we have many things we interact with in our environment that we might not think actually can affect our DNA. So my lab specifically, we look at diet, and even more specifically, we look at one of our favorite things, which is sugar. And we look at how sugar modifies or changes our DNA. And one thing, we looked at trends over time, and we saw that in America, sugar consumption has increased drastically. Obesity rates have increased drastically. And calories consumed in general, the amount of calories the average person eats in a day, have also increased. Now, correlation does not equal causation, but we can still explore how these things might interact with each other. So we noticed this trend, and our lab looks at this. Specifically, we use fruit flies as our model organism. Fruit flies are pretty simple. It's a very common model organism. And what happens is normally things we see in fruit flies, it's simpler circuits, but the same circuits are seen in bigger mammals and in adults. So fruit flies that we put on high sugar diets actually lose the ability to taste sugar over time. I run this assay. It's called proboscis extension response. So this is my fly. It's kind of constrained. And what we do is we actually feed them sugar. And just like a doctor, if they hit your knee, you kind of do this thing. Flies, when they get their taste buds stimulated, they actually stick their proboscis out. And we can quantify this. So this is basically a way to quantify if the fly is tasting the sugar or not. So here we have it. It's like bleh. It's tasting the sugar. Here, we don't get any taste, and then here we get kind of a half response. And what we've discovered is that flies that are put on this high sugar diet, which is this pink diet, at 30% sugar concentration, they taste about the same as flies that are just on any normal diet. At 5% sugar, these flies lose the ability to taste the sugar that the flies that never got put on a high sugar diet can taste. So once again, these flies, the only difference between the two is the ones that are in pink were on a diet high in sugar. The ones that were in black never tasted sugar until we actually did this experiment on them. In addition to just this loss of taste, we also find that these flies actually have increased sugar consumption. So after feeding flies sugar for a week, they eat more and more sugar and more food. And they actually have more fat accumulation as well. So not only are they not tasting the sugar, but they're eating more calories to compensate for this, and they're accumulating more fat. What's really crazy to me, though, is this doesn't appear to be reversible. So if we have a fly on a normal diet, the taste, it's green check mark, looks good. High sugar diet, questionable. If we take this high sugar diet and put it back on a normal diet, it doesn't gain this ability to taste again. It's still stuck like that. So we kind of think of this as like when humans try these new like diet trends and like lose a bunch of weight and gain it all back really quickly. If this is happening in humans, that might not necessarily be your fault if your genome is altered. So we think that an epigenetic modification is happening here. So we think sugar is putting these markers onto your body and making it so that you can't taste. And what I want to do is try to figure out how I can make it so that you can taste again. So what I do is I take a bunch of these different markers, like for example these clips, and I throw them at my fruit flies, and I put them on these markers, and I try to see if there's a way that I can manipulate this taste to get it back to what it should be, what a baseline level is. So I do that through these epigenetic modifications, which we talked about how that environment can influence. I also do this through intermittent fasting, which has been proven in a lot of studies to intermittent fasting means you take them off a diet, you starve them basically, and this has been proven to mess with your feeding physiology. So that's what I do this past summer, and the data we're collecting for that is really interesting, and stay tuned to hear about that. But for the most part, what does this all mean? So you guys heard me blab on a lot about sugar and fruit flies, and first of all, fruit flies are flies, but like I said, these simple circuits seen in these animals can also be seen in humans. We also find that rats also lose this taste. What does this mean for you? Does this mean that, well, our environment and our genes are interacting? It's not one or the other. So you're not prone when you're born to be a sugar addict. It's a combination of the two. Does this mean you should just give up sugar and stop eating sugar altogether? I'm not saying that, but I'm saying be cautiously optimistic. So be optimistic at the fact that we know this knowledge. We know that our environment can have these effects on us. That's something that we didn't know 20 years ago. But be cautious and know that food is fuel for our body, And when we are using bad fuel, don't expect the engine to always be running as best as it can. So thank you guys. Want to say a shout out to my incredible lab mates, all of you in HSF, and all my family and friends for listening to me talk about fruit flies and sugar. Appreciate it.